Multiline variety. Multiline varieties are those variety which is produced from the mixing of several isogenic lines. So before understanding the multiline variety, you should have the knowledge about the isogenic lines, because for the producing of the multiline variety we have some isogenic lines. So isogenic lines, the lines, the genotype, the identical genotype, which is all the characteristics of these plants are same, like height, the leaf pattern, the root length, the yield, all the other characteristics of these lines are same except for the one gene, except for the one character. The differences occur only at a single character, and every character of a living organism is controlled by gene. So isogenic lines are those lines which is identical lines except for the one locus, except for the one gene. The differences possible only at a single gene. So this type of line is called isogenic lines. And this isogenic lines is produced from the plant breeding methods. The name of this plant breeding method that is used for the producing isogenic line is backcross breeding method. Backcross breeding method is highly used in a plant breeding program for the development of disease resistant variety as well as insect resistant variety. When we want to make the disease resistant variety, we have a very good plant breeding method, that is the backcross breeding method is utilized for the producing disease resistant plant. So this isogenic lines is also produced from this backcross breeding method. In case of backcross breeding method, we require the two parent. One is a recurrent parent and other is a non-recurrent parent. This non-recurrent parent is called the donor parent. Donor parent means this parent provide the genes, the disease resistant gene or other genes, the contributing genes. Those parent which contribute the gene is called donor gene, and those parent which intake the gene is called recipient as well as recurrent parent. Recurrent parent means in case of backcross breeding program, we repeated the crossing with the F1 progeny. So the repeated parent is called recurrent parent. So in the other lecture when we making the videos on the backcross breeding method, here I am describing the detailed procedure of the backcross breeding program, but here I am focusing on the concept of the multiline variety, how to produce the multiline variety. So multiline variety is produced from the mixing of the isogenic lines, and this isogenic lines is also produced from the backcross breeding method. And after the making of the isogenic lines, we mixing this isogenic lines the equal amount of seeds of 7 to 8 isogenic lines, and we make the multiline variety. The first idea of the multiline variety was suggested by Janssen in 1952 in the crop oat. And after that, the Norman E. Borlaug has also developed the procedure to making the multiline variety in wheat. The N.E. Borlaug is a famous scientist as well as wheat breeder. He is also known as the father of green revolution involved, N.E. Borlaug. The development of the Mexican dwarf wheat, Triticum aestivum, the credit for the discovery of the Triticum aestivum, the Mexican dwarf wheat, goes to the Norman E. Borlaug. So the multiline variety is a very important type of variety. There are important characteristics featured. The multiline variety, the genetical constitutions of this multiline variety is a homozygous and heterogeneous type of population. The genetic constitutions of this multiline variety is homozygous and heterogeneous type of population. However, in case of pureline variety, the pureline variety is a homozygous and homogeneous type. Homozygous and homogeneous. Homozygous means similar types of alleles are present on the corresponding locus. So in case of homozygous plant, there is no segregation possible. But in case of multiline variety, this is also homozygous. Segregation is not possible. Similar types of alleles are present on the corresponding locus, but it is a heterogeneous type of population, means genetically dissimilar population. How genetically dissimilar population? Because we mixing the seeds of isogenic lines, and these isogenic lines have the single gene difference. All the characters are same. Suppose you have the 8 lines, 8 isogenic lines. All 8 isogenic plants have the similar height, similar yield, all the characters are same, but differences are found only at a single locus. Only one character difference. Because of that, we call the isogenic lines. And this multiline variety is mixing the equal quantity of seeds from these isogenic lines is called multiline variety. So the first feature of this multiline variety, this is the homozygous and heterogeneous type of population. Homozygous means the similar types of alleles are present on the corresponding locus, as well as no segregation happening in the F1 generation. Next is a heterogeneous type of population because genetically dissimilar populations are present. The other important feature of this multiline variety is the higher adaptability, the wider adaptability. This multiline variety grow the different environmental condition, the different types of abiotic condition. This multiline variety survive in the several abiotic stresses as well as environmental factors. Because of that reason, we call the multiline variety has the wider adaptability. The broad genetic base, due to the broad genetic base of the multiline variety, the adaptability of this variety, this multiline variety is more. Wider adaptability means there are 2 types of adaptability. One is the wider adaptability and other is the narrow adaptability. Wider adaptability means the broad genetic base or narrow genetic base. Broad genetic base means this multiline variety is produced from the several lines, several isogenic lines. Because of that reason, this multiline variety has the capacity to survive in the several environmental fluctuations. But in case of pure line variety, this is the narrow genetic base. Why we call the narrow genetic base? Because this pure line variety is the progeny of a single homozygous self-pollinated plant. This is the progeny of a single plant. Because of that reason, we call the narrow genetic base, the genetic transformation from a single plant. But in case of multiline variety, we mixing the seeds from the different isogenic lines. The other very important character is this multiline variety is produced only in self-pollinated crop. This multiline variety cannot be produced in other cross-pollinated crop like maize. This multiline variety is always produced in a self-pollinated crop. So here I am clearing all the things regarding this multiline variety in a very clear manner. So you have to understand this multiline variety in a good way. So generally, this multiline variety is the mixing of the isogenic lines, and this isogenic lines is produced from the backcross breeding methods. Thank you.